Hello everyone. Today we'll be covering small sections of chapter 13, 14, and 19. Just make sure that you notice on the study guide that I'm only covering small pieces or small bits of each chapter. So let's start off with clones. What are clones? And so clones are considered genetically identical molecules, cells, or organisms all derived from a single ancestor. And so in this case, identical twins are also natural clones, clones made by nature, because they are derived from a single ancestral cell. The very first artificial clone was made back in 1997 with a sheep called Dolly. And what was significant about this is that an adult cell was taken from an adult, so from an udder, and was cloned in lab. And so first the cell was then put into an egg cell. So this is the adult cell put into an egg cell. They were fused together. So that means that the DNA was mixed. And so this is, like how this is like artificial fertilization of like sperm and egg, but instead of sperm and egg, it's egg and donor cell. And it was cultured in lab until it turned into an embryo and then re-implanted in the uterus of the foster mother. And the mother was actually able to give birth to a lamb that had the exact same DNA as the donor. And so this is really significant because this showed that even though the cell was already a specialized cell in the adult stage, it could be reprogrammed into cloning an entirely new organism. And of course, this was a big controversial thing, and it still is today. The type of cloning that scientists do now is the cloning of DNA or fragments of DNA, genes, to take the genes from one organism and insert them into a whole different species, and that whole different species can express that gene or into the same species to maybe repair a gene. And eventually, the goal of cloning genes or of DNA is to repair diseases. For example, some people are born with impairments in their insulin genes and can lead to diabetes. So, if there was a possibility to repair that gene before the child is born, that would be a great therapeutic advancement. We don't necessarily do that yet. And I mean, from my understanding, we don't. And we can't, it is difficult to repair genes at the adult stage because remember, you can't just repair a gene in one cell. You have to repair the genes in all of the cells. So if you were to repair a gene, let's say, for example, in an organ like the heart or the spleen, all cells would need to be repaired, and so that brings up like difficulties and issues with that. And so one of the considerations is to clone whole organs from your own cells, and then just to replace your organ. Let's say, for example, if you need organ replacement for whatever reason you may need it. But anyway, going back to cloning, and the type of cloning that is done most often is at the DNA level. And so, in order to clone a piece of DNA, you need three of the following. You need a way to cut the DNA at a specific site to produce consistent, manageable pieces. You need a carrier molecule to hold the DNA for transfer to a host cell. And you need a host cell in which many copies of the selected DNA molecule can be produced. So the way that DNA is cut, and there is such a thing, there's such a way to cut DNA, is by the use of these enzymes called restriction enzymes. And these enzymes are isolated from different types of bacteria. Bacteria do this successfully. And so a very specific type of enzyme, let's say, for example, you use an enzyme called EcoRI or HINE3 or BAM-HI or SAU or HE3, whatever enzyme you choose to use, every single enzyme cuts at a very specific site on the DNA. And so, for example, E. coli can only recognize the sequence GAATTC. And every time it sees a sequence, it'll cut in a diagonal manner. And so it'll always cut the fragments in this way, no other way, and it will not cut anywhere else. And so you can imagine that there's a library of enzymes all with very specific cutting sites. And so we take advantage of this as scientists to cut fragments of DNA and to be able to insert them into a vector. And so this is a vector. And so a vector is the piece of DNA that carries the cloning DNA of interest. And we ligate the gene or the DNA to a vector, and then we introduce the vector with the ligated new gene into a host cell. And so if you notice, you can take the vector, so this is a vector here, which is a piece of circular DNA that is fused together. You can take a restriction enzyme, you can cut it so that it is zigzagged and it creates sticky ends. And you also cut the piece of DNA of interest and you insert it here, and you can create a recombinant DNA. And so the steps of cloning DNA is that the DNA to be cloned is cut with a restriction enzyme. So you take a DNA, you cut it with a restriction enzyme. You take the plasmid vector and you cut it with the same restriction enzyme. Both types of DNA that is cut with this enzyme have complementary sticky ends. When the DNA fragments and the plasmids are mixed, they can be bound together from their sticky ends to form a recombinant DNA. And we can do this by taking advantage of DNA ligase. And DNA ligase is a natural enzyme that we have that can repair DNA by adding back nucleotides and sealing them together. And so after you do this, this is called a recombinant plasmid. And now that you have this vector with this new piece of gene of interest, let's say, for example, you want to introduce an ampicillin resistant gene to yeast or something, you can do that with the recombinant DNA, and you can give it to that yeast, and now that yeast will express that gene that'll make him resistant to ampicillin, and it won't die in ampicillin. So let's take a look at a picture of this. And so here's a DNA of interest. You take a restriction enzyme, and that restriction enzyme cuts it at a very specific site. And then you take the vector, the carrier, which is a circular piece of DNA. You use the same restriction enzyme and you cut as well. And so now both of them, the DNA fragments of interest, so these are the genes of interest, and the vectors, which are the carriers of interest, are both cut with the same enzyme, and now they have sticky ends that will overlap. You take DNA ligase, you put these in a test tube. So you see you put them in a test tube here. You let DNA ligase ligate them together. And now when that circular plasmid reseals, you can introduce that new vector with the DNA of interest into any organism of interest. So let's say, for example, you're introducing it to bacteria. And now that bacteria is expressing that gene that you just gave it. And this is a gene that was foreign to it. It was a gene that it never had before. And now it acquired this whole new gene that it can express. So let's say I'm growing bacteria, and I really want this bacteria to be ampicillin resistant, meaning that if it touches ampicillin, it won't die. It won't kill it, because that ampicillin is used as an antibiotic. And ampicillin can break open the cell wall of bacteria and can kill it. But let's say I want to make these bacteria ampicillin resistant. I don't want them to die in the presence of ampicillin. I can just take that recombinant DNA with the gene that makes it ampicillin resistant. It's with the gene that can fight off the ampicillin. I introduce that recombinant DNA to these bacteria. I grow these bacteria on an ampicillin based media. And so this media here has ampicillin. And you'll notice, just by introducing the gene that makes them ampicillin resistant, is that these bacteria can easily grow on media with ampicillin. And so now I've changed its whole biology. I have it expressing a gene that it normally wouldn't express just by inserting that gene of interest. And so, yeah, we can do this. Scientists are capable of introducing new genes to other organisms. All right, so that was the tidbit of chapter 13 that I wanted you to learn about. That was recombinant DNA plasmids, because chapter 14 talks about biotechnology. And so what is biotechnology? And biotechnology harnesses cellular and biomolecular processes to develop technologies and products that help improve our lives and the health of our planet. And so a piece of biotechnology was those recombinant plasmids, and that's something that's very, very useful. We use it till this day to make many things, including drugs like insulin and stuff like that. And so, what are 5 applications of biotechnology in our lives today? It's to make pharmaceutical products, to treat diseases, for example, with stem cells. Now we're thinking about stem cell research and treating diseases that need stem cell therapy, to produce new varieties of crop plants. And so we can have plants express genes that they have never expressed before just by giving them recombinant DNA plasmids. We can generate animal models for human diseases so that we can better understand how these diseases affect humans. And we can prepare DNA profiles used in forensics and other fields. So this is stuff to solve like crime cases. Going back to the recombinant DNA, one of the most important pieces of biotechnology that we use today. This is yet another picture of recombinant DNA where you take a vector. A vector is also called a plasmid, by the way. You cut out a fragment of the vector, and wherever you cut out the fragment, whatever enzyme you use to cut it out, you also use those same enzymes to cut the gene of interest, the DNA of interest, and then you put it in that place. And so you reanneal it, and now you have a recombinant plasmid. We call it recombination because it's recombining with another foreign piece of DNA. And so this vector here is combining with your gene of interest, and now you can introduce it into a host cell, and that host cell will express the new gene. So we've used recombinant DNA to treat diabetes. So one of the first uses of biotechnology was the production of human proteins for treating diseases. And so insulin is made by recombinant DNA technology. And basically, those plasmids are introduced into bacteria. So these are plasmids that can make insulin proteins. And then we isolate the insulin and we give that insulin as a therapeutic drug. And so, by the way, diabetes is the inability to break down glucose with the use of insulin because the person has very little or lacks insulin. And so, and as a result, a lot of glucose can build up in the cells, and that can be toxic and detrimental. And so, children and adults with type 1 diabetes cannot make insulin, which is a protein that controls blood sugar levels. And if left untreated, diabetes is a crippling disease that kills its victims. And so, before using recombinant DNA to make bacteria produce these insulin proteins, we used to take insulin from animals. And so, as you can imagine, taking insulin from other animals and even from human cadavers and just wherever we could get it, that introduces contamination risks, and as unfortunately has contaminated people when they used to rely on this type of method. But again, now we can just use biotechnology and introduce the insulin gene into bacteria, have the bacteria produce the insulin, then we isolate it and put it, bottle it up, put it in a vial, and now people can inject it. And it's a safe way to gain insulin. And this is human insulin. So we're making bacteria produce human insulin because we gave it the human insulin gene so that it can express it. Biotechnology has also been used to treat hemophilia. Yet this is just another type of disease that can be treated. And so hemophiliacs, they can't make the protein necessary for forming blood clots. And as a result, they can bleed to death. They can have uncontrollable bleeding. And the way that they would get that the blood clot protein was by blood transfusions, except that these people were vulnerable to getting diseases, and when HIV was first around and not really well known, a lot of people ended up contracting HIV just by these blood transfusions. But now, because of biotechnology, because of recombinant DNA, we can now clone the proteins necessary, the clotting factors, which are called like hemophilia A, hemophilia B, and whatnot, with recombinant DNA technology, and we can just have bacteria produce these proteins, and then we can bottle up these proteins and then give it as a therapeutic drug. And so here are some examples of recombinant DNA technology that has been used to make all types of different drugs. So we have something for hypertension and heart failure. We make hormones with recombinant DNA. And we have a hepatitis B vaccine that can be made with recombinant DNA technology, and this is to prevent infection by hepatitis B virus. Human insulin is produced by recombinant DNA to treat diabetes, interferons to treat leukemia, interleukin to treat cancer, and so on. And so, as you can imagine, this type of technology has been very useful in treating diseases. And so we can also employ animals to make these proteins that are then isolated and then given as therapeutic treatment as well. So one example of that is by treating Pompe disease. And so Pompe disease is often a fatal autosomal recessive disorder that weakens and eventually disables heart and muscle function. This disease is caused by a mutation in the gene for acid glucosidase, and it's an enzyme that breaks down glycogen to make it available as energy source for cells. And so if glycogen isn't properly broken down, it can accumulate and then become very toxic to the cells. And so a way to help a person, combat this type of disease is by doing something that's called enzyme replacement therapy. And so this is a treatment in which the enzyme that is necessary, and so we know that the enzyme that the person has is mutated, and so we give them the non-mutated version intravenously, and that serves as a form of therapy. And so now the person has the enzyme that can break down the glucose, and that glucose won't build up as a toxic. And so a way that this enzyme is gained, so this isn't done like by bacteria, this is actually made in hamsters, in animals. Now, why they chose to make it in hamsters and not in bacteria, I'm not sure. But maybe there's some type of issue with making it in bacteria. Maybe it's not as efficient or something, there's just not a good way to do it, because I don't see why they wouldn't choose that route, that they had to employ, animals, to do this. Or maybe it can be with quantity. It could be that making it in animals can produce a larger quantity and maybe cheaper to make. I'm not quite sure on the answer as to why choose animals instead, but I'm imagining it has to do something with quantity. All right, another type of biotechnology that has, to some degree, also caused controversy are stem cells. And so stem cells are the cells that are not specialized yet. So they're not designated to be a heart cell or an eye cell yet, or any other organ, right? A stomach cell, a skin cell. So these stem cells, they have no destination yet. They have no idea what they're going to be yet. So we call them unspecialized cells. And the reason why that's super important is because imagine if you have, let's say, a brain injury or some type of injury in which you need that tissue to be repaired. You're going to employ stem cells, and let's say you have a brain injury, so you can employ stem cells, turn them into neurons, and then replace the site of injury. I mean, that's the theory, right? Not to say that we have the ability to do all of that just yet, although research has advanced pretty far with this. And so a way to gain stem cells is through embryonic stem cells. And so this is during development. So this is like the very first stages of development. And also there are some forms of adult stem cells. Embryonic stem cells form in the blastocyst stage of development in the inner cell mass. So this is at the very beginning of development. These cells, they do not have a destination just yet. And these cells are the most wanted, like the most popular ones, because they're considered pluripotent, meaning that these types of cells can differentiate into any cell type, any tissue, or any organ of the body. And so these pluripotent cells that come from the embryonic stem cells have the capability of becoming any single cell type. That is not the case with adult stem cells. And so, adult stem cells can be recovered from the umbilical cord, blood, also in bone marrow. And these cells, they cannot, they're called multipotent because they can only form a limited number of different types of cells. So they can't just turn into any cell, but we can get a variety of different cell types. And so, because they can't just turn into any cell, they're considered multipotent just because they have limits. But we can still use these type of cells to repair, damaged organs and tissues. And so, why study stem cells? Because the ability to isolate and grow stem cells provides many research opportunities. You can induce them to form different cell types, and in turn, we can use it for tissue repair or even whole organ repair. And so we can also try to understand how diseases work, right? And so we have a stem cell that, it's at its fresh start. We can introduce a disease to that stem cell. And as it's developing, we can understand, okay, why is it that this disease develops? What's going on within the DNA? And so there's a lot of potential with using stem cell research. All right, let's talk about another factor in which biotechnology has played a big role, and this is called GMOs, which you all probably have heard of by now, even the controversies of GMOs, which are genetically modified organisms, but mostly we hear about them when it comes to buying food, buying groceries at the grocery store. And so we've been able to genetically modify crops for thousands of years by artificial selection, meaning that we can cross, two crops with desirable traits in hopes to gain an even more desirable trait in the offspring. That's called artificial selection. But that's not genetically modifying an organism in the way that we do it today, in which we can introduce a whole new gene into an organism that did not have that gene before. And so this is no longer crossing them artificially. This is introducing a new gene and completely changing the way that this organism works, or to an extent, right? Depending on what you introduce. And so genes can be transferred into crop plants using biotechnology by recombinant DNA plasmids, and they're called transgenic plants. And so these transgenic plants are produced faster and more precisely than by plant breeding. And so you have more control over the genes in this case. And so you take a recombinant DNA. Let's say you take a gene from another organism, and it can be from a completely different organism, and you can introduce it into a plant at the embryonic stage. And so when the plant is developing, and now all of the cells in the plant will express that new gene. And so we can make these plants resistant to insects. We can make them resistant to weeds. We can make herbicides with it. And so here are some examples. And so corn, cotton, potato, and tomato, they have been given this new gene. It's an insect-resistant gene. And that insect-resistant gene came from a bacteria. Okay, it came from a soil bacterium. And so now corn, cotton, potato, and tomatoes are resistant to insects, and that just means that we will have more viable crops, so our crops won't just easily die, right, by insects. And so other examples are tomato. We have a gene, delayed ripening, and so that was taken from another tomato that has this trait, and so they got the very best tomatoes, isolated the gene for ripening, and then introduced it to a batch of tomatoes into a new crop. And so the genes can be derived from the same species, right? And so because there's a lot of different alleles in a population, and so not every organism expresses all of the alleles available. And so so these are examples of how we make crop plants more viable, and this is important for us because as our population is expanding, we need to have a better food source, and so making them more viable for growing, is of interest. Except that there's just an issue with genetically modifying organisms, and that's what people call GMOs. And we have an issue with eating these because we're not sure, um, if they're good for us or if they're not good for us, and so there's a lot of controversy with that. All right, now I'm going to switch gears. So now we're going to switch gears from biotechnology to evolution, and I'm only going to lightly touch on evolution just because this book didn't cover too much on it. And so, evolution is basically what happens to our genetics as a population, like as a whole. And so, I'm just going to briefly cover and comment on it. And I assigned some of these sections in chapter 19. Just know that everything that I've covered in my lecture does not include everything that is in the study guide. So make sure that you read through the study guide, that you read the sections that I have covered. You can look at the videos that I have provided as extra resources, but just make sure you dive in a little bit more deeply than what I am covering here. In formulating the theory of evolution, Alfred Russel Wallace and Charles Darwin noticed that some members of a population are better adapted to the environment than others. And they say that these better adapted individuals have an increased chance of surviving and leaving more offspring than those with other genotypes. And so you may have heard of this as survival of the fittest. And the differential survival and reproduction of better adapted genotypes is what they termed as natural selection. So natural selection is basically the surviving alleles in a population that that has survived today, and the allele frequency in that population makes it better adapted than what may have existed before. And so an example of natural selection is the scenario here. And so here you have a bird that has a choice between red bugs and green bugs, but this bird has a preference for green bugs. So what do you think will happen to the population of red bugs over time as these birds prefer the green bugs? So over time, the red bugs will populate, okay? They'll take over the green population until there's way more red bugs than green bugs. And so this is a survival trait. This keeps the bugs from surviving and from not being noticed by the birds. And this is an example of natural selection, where the best fit, in this case the red bugs, because the red bugs are not preferred by birds, these are the best fit, will survive, and that allele frequency, or those alleles, will be the ones that are mostly expressed in that population. And so natural selection is always dependent on the environment. So it depends on the environment circumstances. And so here's another example. In an ecosystem where giraffes have long necks, let's say that there was a situation in their ecosystem in which the shorter shrubs died out. And so let's say this happened, in an area where the shorter shrubs died out and only the longer shrubs or the taller shrubs exist. So which population of giraffes do you think will be best fit to survive in this area? Which ones will natural selection favor? And so if you said the giraffes with the longest necks, you are correct, because the giraffes with the longest necks have a better chance of eating, therefore they're better adapted for survival in this area, just because of the smaller shrubs, that died off. And so in this case, the long-necked giraffes will be the ones that'll be reproducing more, and eventually the population will be more dominated by giraffes with longer necks versus the ones with shorter necks. And this is yet another example of natural selection. And that's what Charles Darwin had noticed, is that there tends to be, in populations, there tends to be alleles that are more frequent than others, because it makes them better adapted for that environment. But should that environment change, the allele frequency will change too. The population, so let's say the environment here changed, and the taller shrubs are not the ones that are, that are most popular in the area. Let's say the shorter shrubs are the ones that are the most popular ones. Well, in this case, both of them will survive equally, because both of them will have a chance of, getting food. And so you wouldn't see one being favored more than the other. Natural selection doesn't necessarily mean that every allele that is surviving in the population is the most favorable allele for that population. In some cases, you may see diseases that are still very persistent. And even though a disease, is detrimental, to people, in some cases can cause death, why would there still be diseases around? Why do they still persist? And so an example is sickle cell anemia, in which cells that are sickled, their whole phenotype is changed. They can't bind oxygen that well, and so if you can't bind oxygen, you can't deliver oxygen that well to the cells, and that causes all sorts of problems. However, there tends to be a trend with the persistence of sickle cell anemia in Africa, and so if you see the map here, there's sickle cell in these areas that are in green and blue, and we also see the same exact persistence of malaria in those same regions. And so, what does malaria have to do with sickle cell anemia? And so, interestingly, people who have sickle cell disease are less affected by malaria. They won't die from malaria because malaria attacks, the blood cells. And so, even though sickle cell disease is not favorable for a human, sickle cell disease still persists in a population simply because people are better adapted to survive malaria. And so malaria is just, one of those killers in Africa. It's a huge disease, it's a huge problem, but yet people with sickle cell tend to die less. And so it makes that disease allele more frequent within that population. And so natural selection doesn't necessarily mean that the allele with the most favorable trait is the one that survives. It's the allele that helps the population produce offspring, that helps them just survive in general, is the one that's or the ones that are most persistent. But then you have other situations, for example, called the founder effect and genetic drift, in which the allele frequency is abundant, not because of natural selection, but mostly because it begins from a small population. And why would you have a small population in the first place? Well, it depends. Some people migrate and live on an island, and then they repopulate, and then whatever allele frequency within that small population is the one that's persistent. And so if there's a disease gene, that disease gene will be persistent within that population. Or there could be something like a natural disaster, like what happened in 1775 in Papua New Guinea. In 1775, there was a huge typhoon that killed 90% of the population, and there were only 20 survivors left. One of the 20 survivors had a disease called achromatopsia, which is a recessive disorder for color blindness. And so, what do you think would happen within a population of 20 in which one individual has this disease? So as the population expanded and expanded and it got bigger and bigger, until real time today, what happened was that this disease here shows up in very high frequency in this population in particular. And that's because the founders, and so this is called the founder effect, and that's because the founders, which were the original ones that carried the disease, were the first ones that started populating the population. And so just as a result, a lot of people ended up with this disease trait simply because they started off from a small population. And so it wasn't necessarily like a natural selection thing. It doesn't help this population, it doesn't give them an advantage in surviving, but yet this allele tends to persist in this population because of what happened, because of the founder effect. And so some races, well, quite a bit of them actually, can be classified genetically simply due to these type of founder effect situations, or due to the fact that there's a certain allele frequency in that population more so than there is in other populations. And so sometimes we can genetically trace what kind of race a person is simply by their genetics and a certain allele frequency that tends to be more populated in one type of race than another. All right, and so that's all that I have to say about chapter 19. I only gave you little tidbits of it just because there's a lot of calculations in there with the little frequencies, but I don't think that's important for this class. However, if you want to read more about it, feel free to. But these were just the little last tidbits and pieces that I wanted to cover from these last chapters, 13, 14, and 19. So after this, we're going to focus on some case studies. I'm going to have you write a paper based on a case study. And then after that, you will take your very last exam, which will cover these previous chapters, and that'll be the end of our course. And so, yeah, this is basically the last study guide that I'll be giving out, and the last quiz based on a study guide. Next week, you'll be receiving your case studies to write your paper.